Imagine for a second that your smartphone um, physically rewired its own motherboard every single month.
Right.
Like picture the processing chip physically swelling in size, the memory drive shrinking, the microscopic wiring pulling tight, and the operating system actually splitting into entirely different temporary software programs.
Right, and then just putting itself back together a few days later.
Exactly. And if you watch that happen, you'd think the phone was completely broken or, infected with some sci-fi virus. But if you are someone who experiences a natural menstrual cycle, this exact biological shape-shifting is happening inside your skull right now.
It really is. And for the longest time, the scientific community essentially looked at that shape-shifting, and while they decided it was just too complicated to deal with.
Yeah.
If we look at the history of clinical neuroscience, the fact that the female brain is this dynamically reorganizing landscape that changes day by day, it was viewed as a nuisance.
A nuisance, wow.
Yeah, it was actually categorized as unwanted variants. Researchers wanted a clean, static baseline, so they overwhelmingly studied male brains or, male animal models. The monthly cycle was treated like static on a radio dial.
But the static wasn't a glitch in the hardware.
Not at all. I mean, modern precision neuroimaging is proving that this cycle is actually a profound, highly sophisticated engine of neuroplasticity. We're looking at a system that doesn't just tolerate change. It's actually designed to continuously optimize its own architecture based on the chemical environment of the moment.
Which is exactly what we are unpacking in this deep dive. We've gathered a stack of incredibly dense, cutting-edge neuroimaging research spanning an entire decade. So from foundational work in 2015 all the way up to the bleeding edge, dense sampling precision imaging papers published in 2025.
It's been a massive decade for this field.
It really has. We're going to translate the neuroscience behind how the menstrual and ovarian cycles profoundly affect the literal structure of your brain, how its networks communicate, and how information flows across it.
Yeah, we're going to look at the actual mechanics of this, like how gray matter physically changes size, how white matter tracks the highways of the brain reorganize their structural integrity, and how the actual flow of information across the brain shifts, borrowing principles from fluid dynamics depending on the day of the month.
Before we jump into the deep end, I want to establish a quick ground rule for how we are approaching the data today. We are going to be talking about how these biological fluctuations relate to very real experiences. So things like mood changes, pain sensitivity, and we will absolutely look at the neurological effects of hormonal contraceptives.
Right.
But our mission here is to impartially report the findings of the original source material. We aren't here to take sides or, endorse any particular medical intervention or political viewpoint. We are strictly looking at the data, explaining the mechanics, and translating the science into something you can use to better understand your own biology.
And knowledge is most valuable when you can actually understand the mechanism behind it. To understand what is happening inside the brain's architecture, we first have to understand the construction crew driving the change. We need to look at the ovarian cycle and the specific hormonal signals that are being broadcast.
Right, because if I break my arm, the bone heals locally. But what we're talking about here is a systemic signal. So what are these chemical signals actually telling the brain to do, and where are they coming from?
To map this out, we can look at a massive foundational systematic review. It was published in 2021 by Dubal and colleagues in the journal Frontiers in Neuroendocrinology. It's titled Neuroimaging the Menstrual Cycle, a multimodal systematic review.
Okay, so a systematic review means they looked at a ton of previous studies, right?
Exactly. They synthesized data from 77 different neuroimaging studies, which encompasses over 1,300 naturally cycling women. So this gives us the definitive baseline for the hormonal landscape.
Over 1,000 brain scans synthesized into one paper.
Mm-hmm.
That gives us a really solid foundation. Walk us through the phases they outlined.
So the cycle operates through what we call the HPG axis. That's the hypothalamic pituitary gonadal axis. It's basically a feedback loop.
Mm-hmm.
The cycle begins with the early follicular phase, which starts with menses.
Right.
During this window, the concentrations of steroid hormones, primarily estrogen and progesterone, are at their lowest. The brain and the ovaries are essentially in a quiet baseline state.
So the chemical construction crew is taking a coffee break.
Temporarily, yes. But then we move into the late follicular, or pre-ovulatory phase, and the biochemical environment radically changes. The brain sends a signal down to the ovaries, and in response, the ovaries begin pumping out estradiol.
Estradiol being a type of estrogen.
Yes, estradiol is the most potent primary form of estrogen in the human body during reproductive years, so it surges to an absolute peak. Meanwhile, progesterone remains flatlined, and right at the very end of this phase, triggering ovulation, we see massive, sharp peaks in two other hormones from the brain:
Which are,
luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH.
I want to pause here because I think a lot of people, myself included, have this mental model from like middle school health class that estrogen and progesterone are just reproductive hormones. We imagine them staying south of the neck, doing things in the uterus and the ovaries. How are they getting up into the brain to cause these changes?
That is a crucial misconception to break. These hormones are highly lipophilic, meaning they are fat-soluble.
Okay.
Because of that, they don't need a special transport mechanism to get into the brain. They just effortlessly cross the blood-brain barrier. The brain is quite literally bathed in them, and once they cross over, they find a brain that is absolutely covered in receptors specifically designed for them.
Wait, so there are estrogen receptors just sitting in regions of the brain that had nothing to do with reproduction?
Everywhere. There are widespread classical nuclear receptors for both estrogen and progesterone, plus membrane-associated receptors in the hippocampus, the amygdala, the prefrontal cortex, the basal ganglia.
Oh.
When estradiol enters a brain cell, it can actually enter the nucleus of the neuron, bind to the DNA, and alter gene transcription. It acts as a powerful systemic modulator of neurochemistry.
So it's not just a floating signal.
Yeah.
It is literally rewriting the instructions inside the brain cells.
Exactly. And following ovulation, the instructions change again. We enter the luteal phase. After the egg is released, the structure it leaves behind in the ovary, the corpus luteum, starts manufacturing massive amounts of progesterone.
Okay, so now progesterone takes over.
Right. Progesterone progressively increases to its peak. Estradiol has a secondary, slightly smaller surge alongside it before both of them fall off a cliff in the last few days of the cycle, which triggers the entire loop to start over.
So if you're listening to this and you have a natural cycle, your brain is currently navigating one of these chemical tides. But let's get into the physical reality of what that tide actually does. If estradiol is getting into the nucleus of a brain cell and changing the instructions, what happens to the physical hardware of the brain?
It shapeshifts. We can see this physical reconfiguration in the gray and white matter. Let's start with a landmark paper from 2015 by Lisovsky and colleagues. It was published in Neuroimage, titled Hippocampal Volume and Functional Connectivity Changes During the Female Menstrual Cycle.
The hippocampus. That's memory, right?
Yes, it's the brain's primary center for learning, memory, and spatial navigation. They used a technique called whole brain voxel-based morphometry.
Voxel-based morphometry. Let's translate that. A voxel is like a three-dimensional pixel, right? Kind of like a block in Minecraft.
That's a great way to visualize it, actually. When researchers do an MRI, they break the image of the brain into millions of these microscopic 3D blocks, or voxels. By measuring the density and volume of tissue within every single voxel, they can see exactly where the brain is physically expanding or contracting.
Okay.
Lisovsky's team scanned 21 naturally cycling women at 4 different time points across their monthly cycle.
And what did the voxel show?
They found bilateral increases, meaning on both the left and right hemispheres, in hippocampal gray matter volume, specifically during the late follicular phase.
Which, if we look at our map, is exactly when estradiol surges to its peak right before ovulation. So the memory center of the brain physically swells in size. But hold on, if my brain's gray matter is literally swelling, wouldn't my skull feel tight? Like, are we talking about the brain sloshing around, or is it just retaining water weight?
That is exactly the question the researchers asked. To figure it out, they didn't just measure the overall size. They used diffusion-weighted imaging to look at what water molecules were doing inside that specific tissue. And they found that as the gray matter volume went up, the mean diffusivity went down.
Okay, mean diffusivity. Break down the physics of that for us.
Think of a sponge. If a sponge just swells up with pure water, the water molecules inside have plenty of room to bounce around in random directions. That is high mean diffusivity.
Done.
But if you take that sponge and you start packing it full of solid material, say, you start weaving thick threads of cotton through all the holes the water molecules get trapped, their movement is restricted. That is low mean diffusivity.
I see. So because the diffusivity went down, the hippocampus wasn't just bloated with fluid. It was actually packing itself with new, solid biological material.
Precisely. Estradiol has a profound neurotrophic effect.
Neurotrophic.
It essentially means growth promoting. When estradiol binds to the receptors in the hippocampus, it triggers mechanisms like synaptogenesis. That's the actual physical creation of new dendritic spines, which are the connection points between neurons.
So new neural pathways are being built.
Yes. It also triggers angiogenesis, which is the creation of new microscopic blood vessels to support that new neural infrastructure.
That is incredible. Over the course of just a couple of weeks, your brain is physically constructing new microscopic bridges and supply lines in its memory center, driven entirely by an estrogen surge.
It is.
But what happens when the tide turns? Like, what happens when progesterone takes over in the luteal phase?
Well, progesterone often acts as a counterbalance, but it also drives its own unique structural changes. While estradiol is busy plumping up the hippocampus, progesterone targets different real estate. A 2018 paper by Pletzer and colleagues in Scientific Reports titled Subcortical Structural Changes Along the Menstrual Cycle. Beyond the hippocampus, they looked at a larger sample of 55 women.
And what did they find?
They found a significant progesterone-dependent increase in gray matter volume in the right basal ganglia during the luteal phase.
The basal ganglia. That's deep in the subcortical region of the brain. What does that area actually do?
So the basal ganglia is a complex network of structures primarily involved in motor control, but it is also deeply tied to reward processing, motivation, and emotion. What is really crucial about Pletzer's finding is that this structural swelling was lateralized to the right hemisphere.
Why does the right side matter so much?
Because reductions in right basal ganglia volume are a known neuroanatomical marker in mood disorders, like unipolar and bipolar depression.
Oh, wow.
Yeah. The authors of the study speculated that as progesterone peaks in the mid-luteal phase, it forces this structural growth. But as progesterone rapidly drops off right before menses, that volume might quickly reduce. That rapid structural contraction in the reward and emotion center might mediate the hormone-related mood swings many people experience in the days right before their period.
So PMS mood swings aren't just some abstract emotional fragility. They could literally be the psychological side effect of a major emotional processing hub deep in your brain, rapidly losing physical mass as the hormone serotonin drops off.
Exactly.
That completely changes how you view a mood swing.
It really grounds the psychological experience in a physical reality, and this connection between physical brain structure and your lived experience becomes even more obvious when we look at the outer layer of the brain, the cortex.
All right, let's talk about the Meeker 2020 paper, because this one literally blew my mind when I read the sources. This paper looked at how the thickness of the cortex changes, and they actually tied it to physical pain.
Yes. Meeker and colleagues published a fascinating study in 2020 titled Menstrual Cycle Variations in Gray Matter Volume, White Matter Volume, and Functional Connectivity. They looked at cortical thickness and found that a region called the left inferior parietal lobule is at its absolute thickest during the menstrual phase itself. But the finding that is most relevant to the listener's daily life involves the somatosensory cortex.
The somatosensory cortex. For the listener, this is the strip of brain tissue that processes touch, temperature, and pain from all over your body.
Correct. The researchers found that the gray matter of the somatosensory cortex was actually at its thinnest during the follicular phase, which is when hormones are relatively low and just beginning to rise.
Okay, so the pain processing center is physically thinner. But they didn't just scan these women's brains. They actually hooked them up and tested their sensitivity to electrical pain, right?
They did. They applied quantitative sensory testing, which involves delivering safe but measurable electrical impulses to the participants to find their pain thresholds.
Yeah.
And the results perfectly mirrored the structural data. During the follicular phase, when that specific cortex is physically thinnest, the women reported the highest electrical detection sensitivity. They felt the electricity sooner at lower voltages.
Wait, I need to make sure I'm understanding this because this is huge. You're saying that the physical thickness of the part of my brain that processes touch actually shrinks and expands over the month. And when it shrinks, my literal threshold for feeling an electric shock goes down.
Yeah.
I become more sensitive to physical stimuli because my brain tissue is thinner.
That is what the data indicates. The structural variance directly translates to sensory variance. It's a brilliant illustration that these hormonal updates are not just invisible background processes. They literally dictate how intensely you physically collide with the world around you.
I can't even imagine how validating that data is for someone who deals with chronic pain that flares up at certain times of the month. It's not in your head. Well, I mean, it is in your head, literally, but it's a structural reality.
Physical.
So we've got the memory centers growing, the emotion centers swelling and shrinking, the pain receptors thinning out. What about the connections between all of these? The white matter.
White matter is essentially the brain's cabling system. It consists of bundled axons coated in myelin, which allows electrical signals to jump rapidly across long distances. And yes, the cabling changes too. A 2023 paper by Reiser and colleagues titled Menstrual Cycle-Driven Hormone Concentrations Co-Fluctuate with White and Gray Matter Architecture Changes Across the Whole Brain.
That is quite the title.
It is, but it demonstrated something amazing. It showed that these hormone concentrations co-fluctuate with white matter microstructural anisotropy.
Okay, anisotropy. I'm going to need you to unpack that one.
Imagine a bundle of optical cables. If all the cables are perfectly straight, tightly bundled together, and the light is shooting purely in one specific direction, that is high anisotropy. It's highly directional and highly efficient.
Okay.
If the cables are frayed, loosely packed, or crossing over each other, the signal diffuses in multiple directions. That's low anisotropy.
So high anisotropy means the neural highway is paved perfectly smooth, straight, and fast.
Precisely. And the Reiser paper showed that when estradiol, LH, and FSH hit their peaks right around ovulation, white matter microstructural anisotropy increases across the whole brain.
Wow.
The entire communication network tightens up and becomes highly directional. Conversely, in the luteal phase, when progesterone peaks, they saw an increase in mean diffusivity in the white matter, which suggests a slightly looser, perhaps more winding or diffuse structural state.
So estrogen acts like a microscopic construction crew, pulling all the cables tight, optimizing the whole system for pure speed and efficiency. And just to bring this all the way up to the present day, what is the absolute latest science saying about this? Because this field seems to be moving incredibly fast.
It is, largely because of a technique called dense sampling. Historically, we'd scan a group of women once or twice a month and average the results. But in 2025, Heller and colleagues published a landmark paper called Hormonal Milieu Influences Whole Brain Structural Dynamics Across the Menstrual Cycle Using Dense Sampling.
Dense sampling. What does that mean in this context?
Dense sampling means they took the same individuals and put them in an MRI scanner over and over again many times across a single cycle.
So scanning the exact same brain day after day to watch the shape-shifting happen in real time.
Exactly. And what Heller's 2025 data proved is that these structural volume changes are not just happening in isolated pockets. It's not just the hippocampus here or the basal ganglia there. It is a highly coordinated spatiotemporal pattern of brain volume changes distributed across the entire brain.
The whole thing is moving.
The whole organ is engaged in a synchronized structural dance. And in a naturally cycling individual, this widespread morphological dynamic tracks incredibly strongly with serum progesterone levels.
Now, I have to ask the question that is probably on the mind of a large percentage of our listeners. What happens to this synchronized whole brain dance if you introduce oral contraceptives?
That is a critical variable, and we have to look at it objectively through the data.
Yeah.
Heller's 2025 study specifically included cohorts of individuals taking oral contraceptives, as well as those with endometriosis.
And what did they find?
The findings were stark. In individuals taking oral contraceptives, the typical structural dynamics that widespread progesterone-driven shapeshifting was fundamentally altered. Instead, any structural variance tracked primarily with the exogenous estradiol provided by the pill.
So when you suppress the natural endogenous hormone fluctuations to prevent ovulation, you also suppress the natural neuroplasticity engine. Like the brain stops shapeshifting in the same way.
The natural intricate dance between estradiol and progesterone is overridden by a steady state of synthetic hormones, which stabilizes the physical architecture but alters its natural plasticity.
Okay, so let's summarize where we are for the listener. Depending on the week of the month, your hippocampus might be physically larger, sprouting new connections. Your basal ganglia might be swelling. The cortex where you process pain is physically thinning out, making you more sensitive. And the white matter highways connecting all of this are pulling tight and becoming super efficient.
That's the structural picture, yes.
All of this physical hardware is changing. But, I mean, if my laptop's motherboard physically warped and changed shape while I was typing an email, the software would crash. How does the brain software, the actual functional communication between these regions, not completely short-circuit when the hardware changes?
That is the perfect transition because the brain doesn't just survive the structural changes. It leverages them to change how it processes information. Let's move from structural plasticity to functional connectivity, basically how the wiring actually fires.
Let's do it. When we talk about functional connectivity, we're usually talking about resting state fMRI, right?
Yes, resting state functional MRI measures spontaneous low-frequency fluctuations in brain activity when a person is just lying in the scanner, not doing any specific
So
task.
it's resting.
Right. By seeing which regions light up at the same time, we can tell which areas are communicating. We can see the networks.
Who is on a conference call with who, and who is the boss of the call?
That's a great way to put it. And a 2020 paper by Hidalgo Lopez and colleagues in Brain Structure and Function titled Human Menstrual Cycle Variation in Subcortical Functional Brain Connectivity, a multimodal analysis approach. They utilized a multimodal analysis to look specifically at subcortical connectivity.
And what did they see?
They found that during the pre-ovulatory phase, when estrogen is skyrocketing, there is a massive increase in frontostriatal connectivity in the right hemisphere.
Frontostriatal. So that's the connection between the frontal lobe, the CEO of the brain, making executive decisions, and the striatum, which handles reward, motivation, and habit formation.
Yes, the CEO is suddenly in constant direct communication with the motivation department. But it gets even more interesting in the luteal phase, when progesterone takes over. Hidalgo Lopez found that during this phase, the hippocampus exhibits heightened eigenvector centrality.
Okay, eigenvector centrality. We are throwing a lot of network science terms around today. Let's ground this. What does that actually mean?
Let's use a social network analogy. If you have a lot of followers on social media, you have high degree centrality. You just know a lot of people. But eigenvector centrality is different. A region with high eigenvector centrality isn't just connected to a lot of regions. It is connected to other regions that are themselves highly connected.
Oh, I see. It's like being the ultimate influencer.
Yeah.
You might not know everyone in the room, but you have the direct cell phone numbers
Yeah.
of the 5 most powerful people in the room.
That is an excellent analogy. During the high progesterone luteal phase, the hippocampus, the memory and spatial navigation center, becomes the ultimate neural influencer. Its functional importance to the entire global network skyrockets.
Which makes sense,
Yeah.
because as we learned earlier, the hippocampus is packed with progesterone receptors. But the network changes get even wilder when we look at the speed of the brain. Because fMRI is great, but it's kind of slow, right? Like it measures blood flow, which takes a few seconds to change.
Right. If you really want to see the brain's topology shift in real time, you need magnetoencephalography, or MEG. MEG measures the tiny magnetic fields generated by the actual electrical firing of neurons. It operates on a scale of milliseconds.
Milliseconds, wow.
Yeah. In 2020, Liparodi and colleagues published a study in the Journal of Neuroscience Research titled Functional Brain Network Topology Across the Menstrual Cycle is Estradiol Dependent. They used MEG to map functional brain network topology across the cycle.
And what did they find when they looked at the magnetic fields?
They discovered that during the periovulatory phase, right around ovulation, the global network integration actually reduces slightly, but simultaneously, the betweenness centrality of a specific region called the right posterior cingulate gyrus significantly increases.
Betweenness centrality. Okay, so if eigenvector centrality is being the ultimate influencer, what is betweenness centrality?
Betweenness centrality is about being the ultimate bridge, or the critical bottleneck. If a region has high betweenness centrality, it means that the shortest path of communication between many other brain regions has to route directly through it. It acts as the grand central station of the network.
So right around ovulation, this one area, the posterior cingulate gyrus, suddenly becomes the grand central station that all the magnetic electrical traffic has to pass through. Do we know what that actually feels like for the person? Like, does it change their psychology?
This is where the Liparodi paper is truly brilliant. They didn't just scan the brain, they gave the participants a battery of psychological assessments, and they found that this specific topological change, this increase in betweenness centrality driven by estradiol, correlated positively with the participants' scores on a psychological metric for environmental mastery.
Environmental mastery. Well, let me guess. That's the feeling of being completely competent, in control, and able to manage whatever your environment throws at you.
Exactly. The feeling that you have agency and capability. When estrogen spikes, this specific gyrus becomes the brain's massive routing hub, and psychologically you feel an elevated sense that you are crushing it. You are mastering your environment.
I love that. The structural bridge physically manifests as a psychological feeling of control. But as amazing as that is, I want to move to the paper that completely shattered my understanding of neuroscience.
Yeah.
The 2021 Miller paper. The 28andMe project.
Yes. Published in the journal Network Neuroscience, titled Dynamic Community Detection Reveals Transient Reorganization of Functional Brain Networks Across a Female Menstrual Cycle. This is arguably one of the most important dense sampling studies ever conducted on the female brain.
Explain the setup of 28andMe, because the dedication required for this study is wild.
It was a massive undertaking. The lead researcher actually scanned a single, healthy female participant who was naturally cycling for 30 consecutive days.
30 days.
Every single day she went into the fMRI scanner, and every single day they drew blood to precisely track her estradiol, progesterone, LH, and FSH levels. They generated a completely unprecedented day-by-day map of functional brain networks.
30 days in an MRI tube. That is a hero of science right there.
Yeah.
What were they looking for with all that data?
They applied a complex mathematical framework called dynamic community detection. Essentially, they wanted to see how the brain's various functional networks grouped together, communicated, and evolved over time. Normally, we think of brain networks as fairly static. The visual network talks to the visual network. The motor network talks to the motor network.
Right, and what did the 30 days of data reveal?
For most of the month, that assumption held true. The brain's major networks, the visual, somatomotor, control, and the default mode network were highly stable. But during the narrow ovulatory window, driven by that sharp combined peak in estradiol, LH, and FSH, well, something incredible happened. The default mode network literally split apart.
Okay, I have to push back on this because when I read that, I was genuinely alarmed. The default mode network, the DMN, is basically the foundation of our internal psychology. For the listener, the DMN is the massive network that is active when you are daydreaming, when you're thinking about your past, planning your future, or reflecting on your sense of self. It is your internal resting heartbeat. Are you telling me that an estrogen spike physically breaks this core network apart? How does a person not feel completely mentally fragmented or dissociate when that happens?
It's a completely rational reaction to hear network splitting and assume it's a pathological breakdown, but in this context, it is the exact opposite. It is a feature, not a bug. The researchers described it as a transient state of remarkably high network flexibility.
Flexibility.
Right. What happened was that the prefrontal nodes of the DMN, the parts residing in the front of the brain, temporarily severed their standard connections with the rest of the DMN and formed a newly transient, independent functional community.
So they didn't break. They just formed a highly specialized temporary task force.
Precisely. They dynamically reorganized to increase their communication with other networks. The limbic, subcortical, and attention networks also showed higher than expected levels of flexibility during this exact same window. The brain was prioritizing between network integration over standard, rigid network isolation.
So for a window of a few days around ovulation, the whole brain just becomes this incredibly adaptable, fluid, cross-communicating ecosystem. The boundaries between departments dissolve so everyone can share information.
Yes. And what is critical to understand is that this massive reorganization subsided immediately after the ovulatory window closed. The DMN reformed, the standard boundaries returned. It was a temporary, localized pattern of radical reorganization driven entirely by the endocrine system's peak chemical output.
That completely changes how we should think about cognitive adaptability. But again, I have to bring up the variable of oral contraceptives because the Mueller study looked at that too, didn't they?
They did. And we must look at the findings impartially. The 28andMe project wasn't just 30 days. It was actually an extended longitudinal study that captured data under different conditions. When the participant was on a hormonal regimen that suppressed ovulation, meaning the massive peaks of estradiol, LH, and FSH were flattened out, that transient reorganization completely disappeared.
The DMN didn't split.
No. The network flexibility was entirely suppressed. The brain's major networks remained highly stable and rigid across the entire 30 days. When we compare this functional data from Mueller in 2021 with the structural dense sampling data from Heller in 2025, a very clear, objective picture emerges. The natural cyclical fluctuations of endogenous hormones serve as a profound catalyst for both structural and functional neuroplasticity.
But when you flatten the hormones,
You stabilize the brain, but you also silence that specific rhythm of network flexibility.
it's an incredible trade-off that millions of people make, often without ever being told that it affects anything above the neck. We've covered the localized structural changes, and we've covered the specific network rewiring. But let's zoom out to the absolute highest level. Let's look at the brain as a single, holistic, machine processing reality.
To do that, we have to borrow mathematical models from physics. Which brings us to a 2021 paper by DeFilippi and colleagues. It was published in Frontiers in Neuroscience, titled "The Menstrual Cycle Modulates Whole Brain Turbulent Dynamics."
Turbulent dynamics. When I hear turbulence, I think of an airplane hitting rough air and bouncing around. is the brain bouncing around?
It's actually a concept borrowed from fluid dynamics, not aerodynamics. Think of a rushing river instead of an airplane. In fluid dynamics, turbulence describes the highly complex, chaotic, multi-scale flow of energy. If water is flowing perfectly smoothly in straight lines, it's called laminar flow. It's calm, but energy and information don't mix very well between the left bank and the right bank.
Okay, so laminar flow is calm water.
Right. But if you introduce boulders and steep drops, you get turbulence, rapids. In rapids, energy is constantly swirling, mixing, and transmitting across the entire width of the river.
Ah, okay.
In the human brain, turbulence is a mathematical framework used to measure how information is transmitted across different spatial scales. It measures how well local millimeter-sized brain regions can synchronize to send highly complex information across long distances, say, from the very back of the brain all the way to the front.
So a highly turbulent brain is actually a highly efficient, intensely communicative brain. The rapids are a good thing.
Exactly. High turbulence allows for rapid, complex information transfer. By analyzing whole brain fMRI data, DeFilippi's team discovered that the luteal phase, the phase dominated by high progesterone, is characterized by significantly higher information transmission across spatial scales than the follicular phase.
Wow. So estrogen might pave the local neural highways and split the DMN into task forces, but progesterone turns the entire continent into a churning, highly complex network of long-distance communication.
That is a phenomenal synthesis of the two phases. During the luteal phase, there is heightened turbulence specifically involving the default mode, salience, and control networks. The whole brain is engaging in highly complex, long-range information transfer.
You just paint such a dynamic picture.
Yeah.
But how does this all tie together? Is there a master study that looks at everything, estrogen, progesterone, structural shifts, and fluid dynamics all at once?
There is, and it is the most recent comprehensive analysis we have. A massive 2024 paper published in the Nature Partner journal NPJ Women's Health by Avila Varela, Hidalgo Lopez, and their extended team. It is titled Whole Brain Dynamics Across the Menstrual Cycle: The Role of Hormonal Fluctuations and Age in Healthy Women.
What makes this 2024 study the holy grail of this topic?
Scale and methodology. Instead of looking at one person for 30 days or looking at just one isolated network, they looked at 60 healthy, naturally cycling women across 3 highly specific hormonal windows.
Which were?
The early follicular baseline, the pre-ovulatory estrogen peak, and the mid-luteal progesterone peak. And they applied a mathematical framework called intrinsic ignition to measure a property of the whole brain called node metastability.
Okay, node metastability. That sounds like something from a quantum physics textbook.
Oh, that's...
I need an analogy for this one.
Let's use music. Imagine a brain region with low metastability as a musician who only knows how to play one rigid piece of classical sheet music over and over again. They play it perfectly, but they can't deviate. They are locked into one state.
Very rigid. Not very adaptable.
Right. Now, imagine a brain region with high metastability as a master jazz musician. They aren't locked into anything. They can fluidly jump from a rapid piano solo to backing up the saxophone to dropping the tempo entirely without ever missing a beat. They can spontaneously coordinate with anyone on the stage.
That's a great image.
In neuroscience, metastability is a measure of dynamical complexity. A highly metastable brain region can rapidly switch its activity and communicate seamlessly with many different networks depending on what the environment demands.
Okay, I love that. A highly metastable brain is a nimble, adaptable jazz band. So what did the 2024 study find when they looked at the 60 women? Which phase produces the best jazz musicians?
The results were definitive. Across the entire whole brain functional network, the pre-ovulatory phase, when estradiol surges to its absolute peak, exhibited the highest global dynamical complexity. The brain was at its absolute most metastable.
Okay.
The mid-luteal phase, driven by progesterone, was the second highest, and the early follicular phase, when all hormones are quiet, had the lowest dynamical complexity.
So estrogen acts as the ultimate catalyst for brain adaptability. It turns the entire brain into a master improviser.
Yes, but Avila Varela's team ran highly nuanced multi-level models, and they found that the roles are beautifully complementary. Higher estradiol specifically predicted higher metastability in the limbic network, the dorsal attention network, and the subcortical networks. It makes you highly adaptable in how you pay attention and process emotions.
And what about progesterone?
Higher progesterone specifically predicted higher metastability in the default mode network. It keeps your internal self-reflective network highly nimble.
That is just, I mean, it's a biochemical symphony. Estrogen comes in, and hypes up your attention and emotional flexibility, and then progesterone tags in and keeps your internal sense of self fluid and adaptable. And didn't this study also look at what happens as women get older?
They did, which was a brilliant addition. They looked at the effect of advancing age within this young adult cohort, meaning as women move from 18 toward 35 years old, and they found that advancing age actually increased whole brain metastability. The brain's dynamical complexity generally increased as these women got older, specifically in the control and dorsal attention networks.
Wait, really? We usually associate aging with a loss of neuroplasticity, a loss of flexibility. But you're saying that as these women progressed through their 20s and early 30s, their brains actually became more dynamically complex.
Yes, the brain learns how to play jazz better the longer it practices. The repeated, cyclical exposure to these massive hormonal tides year after year might actually train the brain to become more functionally flexible and resilient over time.
So synthesizing this all together, estrogen drives peak complexity, structural growth in the memory centers, and even splits the DMN to handle specialized tasks. Then progesterone comes in, swells the reward centers, and drives distinct long-range turbulent connectivity, keeping the DMN highly metastable.
Exactly.
The female brain is constantly dynamically shifting its entire information processing strategy to optimize for its current chemical environment. It is not a static machine, it is a wildly adaptable ecosystem.
You've captured the exact paradigm shift that modern neuroscience is currently undergoing.
Which brings us to the final and frankly the most important part of this deep dive. We spent an hour talking about voxels and mean diffusivity and dynamic community detection. But why does all of this dense, incredibly complex neuroscience matter to the listener right now?
It matters because it finally provides the underlying objective mechanical explanation for their lived experience. For generations, people who menstruate have reported cyclical changes. They report changes in pain perception, changes in cognitive clarity, shifts in memory recall, and profound changes in mood.
Right.
And for generations, the medical establishment often dismissed these reports as subjective, exaggerated, or purely psychological.
The classic, deeply frustrating diagnosis of, well, it's all in your head.
Exactly. And the profound irony of this research is that, yes, it is literally in your head, but not in the dismissive, imaginary way it was meant. It is in the physical structure and the topological wiring of your brain tissue. The cyclical changes in your pain perception aren't imagined. Your somatosensory cortex is literally physically thinning and thickening over the month.
Your pain threshold is physically changing.
Yes. The cognitive flexibility and those days where you feel a surge of environmental mastery aren't coincidence. Your right posterior cingulate gyrus is physically acting as a superhub for whole brain magnetic communication. The mood swings before your period aren't just you being overly emotional. The deep reward and emotion centers of your basal ganglia might be physically shrinking as progesterone drops.
It completely reframes how we view the menstrual cycle.
Yeah.
We are taught in middle school health class that this cycle is purely a reproductive function. Its only purpose is to prepare the uterus for an egg, and if there is no egg, you bleed and start over. But looking at a decade of MRI data, the menstrual cycle is actually a profound systemic neuromodulatory rhythm. It is a brain cycle just as much as it is a reproductive cycle.
That is the crucial takeaway. The healthy brain's ability to adapt to these acute physiological changes is a testament to its resilience. We frequently talk about the triple network model in neuroscience that comprises the central executive network, the default mode network, and the salience network. These three networks dictate how we think, feel, and act, and all three undergo profound regulation by these cyclical hormones.
But what happens when the ecosystem struggles? Like, what happens if the brain can't adapt to that monthly software update smoothly?
This is where the clinical applications of this research become so vital. As Hidalgo Lopez pointed out in her 2020 paper, the fluctuations we see in resting state activity reflect the healthy brain's ability to adapt. The lack of such adaptive process is an inability of the brain's network to flexibly reorganize when the hormones surge or drop well that could make the brain highly vulnerable to pathology.
Meaning the brain gets stuck.
Precisely. If the hormones surge, but the brain's topology remains rigid, it causes friction. This friction could be the underlying mechanical etiology behind why some individuals suffer from severe conditions like premenstrual dysphoric disorder or PMDD.
Oh, wow.
Their networks might literally be struggling with the turbulent transitions. They aren't reaching the necessary states of measurability to handle the chemical tide.
So by mapping the healthy shape-shifting brain, we finally get a baseline. We can stop treating menstrual cycle-associated mood disorders as mysterious hormonal curses and start treating them as specific network adaptability issues. We can look at the brain circuitry and say, oh, the DMN isn't splitting the way it should, or the basal ganglia isn't swelling.
Exactly. It validates the biological reality of the experience and offers a genuine roadmap for targeted neurological intervention in the future.
Okay, let's take a deep breath and recap the unbelievable journey we just took through the human brain. We started with the physical hardware. The gray matter literally blooming and creating new synapses in the hippocampus on an estrogen surge. We saw the right basal ganglia swelling on progesterone and the pain cortex thinning out to make you more sensitive to the world. We watched the white matter highways tighten into straight, efficient cables and then diffuse into winding roads.
We then moved up a level to the software, watching the default mode network literally break apart into transient, specialized task forces during ovulation, and the posterior cingulate gyrus become the brain's central command station for feeling like you've mastered your environment.
And finally, we zoomed all the way out to see the entire brain functioning as a turbulent, rushing river of information transfer, reaching peak dynamical complexity, acting as a master jazz musician right at the crest of the hormonal wave.
It is a masterful, continuously adapting biological system.
Which leaves us with a final, provocative thought. As we discussed at the very beginning, historically, clinical neuroscience defaulted to studying male brains, or male animal models. The hormonal fluctuations of the female menstrual cycle were deemed too noisy. They were considered a nuisance that would mess up the clean, static data sets. Science wanted an unchanging, stable machine to study.
It was a bias for artificial stability over biological reality.
But considering that this noise, this massive monthly fluctuation, is actually a highly sophisticated, naturally occurring engine of neuroplasticity and whole brain network reorganization. Well, what if science has spent the last century ignoring the very biological blueprint for brain resilience and adaptability that we've been searching for all along? What if the noise they were trying to filter out was actually the most important signal the human brain produces?
That is a paradigm-shifting question, and one that should fundamentally change how every neuroscientist approaches their data.
I encourage you, the listener, to observe your own rhythms this month with this new scientific lens. When you feel a shift in your memory, your mood, or your sensitivity, remember that you aren't just experiencing a reproductive cycle. Your brain is actively, physically resculpting itself. Thank you for taking this custom-tailored deep dive into the literature with us. We'll see you next time.